I bet you didn't know that the blueprint of life was discovered in a pus-soaked bandage. In 1869, a Swiss chemist named Friedrich Miescher was hard at work in his laboratory. He was studying white blood cells, which he painstakingly extracted from pus-soaked bandages. Miescher wasn't interested in infections, though. He was after something far more fundamental, the building blocks of life itself. Miescher discovered a substance inside the cell's nuclei that was unlike anything known at the time. It was acidic and rich in phosphorus, which was unusual for organic molecules. He called it nuclein, unaware that he had stumbled upon the very blueprint of life, deoxyribonucleic acid, or DNA. Miescher's discovery was met with indifference. The scientific community, fixated on proteins as the carriers of heredity, largely ignored his findings. Little did they know that this nuclein held the key to understanding life's grand design, hidden within its enigmatic structure. The world would have to wait several decades for the true significance of Miescher's discovery to come to light. His nuclein would become the foundation for a scientific revolution, one that would transform our understanding of life itself. Decades after Miescher's discovery, scientists began to unravel the secrets of DNA's structure. They knew it was a long, chain-like molecule made up of repeating units called nucleotides. Each nucleotide contained a sugar molecule, a phosphate group, and one of 4 nitrogenous bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These 4 bases, like letters in an alphabet, held the key to DNA's ability to store and transmit genetic information. But how were they arranged, and how did this arrangement dictate the diversity of life? The race to decipher the structure of DNA intensified in the early 20th century. Scientists used X-ray diffraction to study DNA's shape, revealing tantalizing clues about its helical structure. But the precise arrangement of its components remained a mystery. The answer, when it finally arrived, would come from an unlikely duo working at the Cavendish Laboratory in Cambridge, England. Their names were James Watson and Francis Crick, and their discovery would change the world. In 1953, James Watson and Francis Crick achieved what many thought impossible. They solved the puzzle of DNA's structure. Their model, the now iconic double helix, resembled a twisted ladder. The sides of the ladder were formed by alternating sugar and phosphate molecules, while the rungs were made up of the nitrogenous bases. But the true elegance of Watson and Crick's model lay in its revelation of base pairing. Adenine always paired with thymine, AT, and guanine always paired with cytosine, GC. This complementary base pairing explained how DNA could replicate itself with remarkable fidelity, ensuring the accurate transmission of genetic information from one generation to the next. The discovery of the double helix sent shockwaves through the scientific community. It provided a physical basis for heredity and opened up new avenues of research in genetics, molecular biology, and medicine. Watson, Crick, and Maurice Wilkins, whose X-ray diffraction images were crucial to their discovery, were awarded the Nobel Prize in Physiology or Medicine in 1962. With the structure of DNA deciphered, the next challenge was to crack its code. Scientists knew that the sequence of bases in DNA held the instructions for building and maintaining an organism. But how did this 4-letter alphabet translate into the complexity of life? The answer lay in the concept of the genetic code. Scientists discovered that a sequence of 3 DNA bases, called a codon, coded for a specific amino acid, the building blocks of proteins. Proteins, in turn, were the workhorses of the cell, carrying out a vast array of functions. The genetic code was found to be nearly universal, meaning that the same codons coded for the same amino acids in almost all organisms. This finding underscored the fundamental unity of life on Earth, suggesting that all living things shared a common ancestor. Understanding the genetic code was a monumental achievement. It provided the Rosetta Stone for deciphering the language of life, enabling scientists to study genes, their functions, and their role in health and disease. Section 5. The Human Genome Project, Mapping Our Genetic Blueprint. In 1990, an ambitious international effort known as the Human Genome Project was launched. Its goal was to map the entire human genome, the complete set of genetic instructions found in a human cell. This monumental task involved sequencing the 3 billion base pairs that make up our DNA, a feat that required cutting-edge technology and international collaboration. The Human Genome Project was completed in 2003, marking a turning point in our understanding of ourselves. For the first time, we had a comprehensive blueprint of our genetic makeup, providing unprecedented insights into human evolution, disease, and the very essence of what makes us human. The project's impact on medicine has been profound. It has facilitated the identification of genes associated with various diseases, paving the way for new diagnostic tools, gene therapies, and personalized medicine. The Human Genome Project was just the beginning. It ushered in a new era of genomic research, leading to the development of faster, cheaper, and more powerful sequencing technologies. These advances have further accelerated our understanding of genetics and its implications for human health. Section 6: Genetic Engineering, the dawn of a new era. The ability to read and understand DNA sequences led to another groundbreaking development: genetic engineering. Scientists could now manipulate DNA in the laboratory, inserting, deleting, or altering genes to study their function or create organisms with desired traits. One of the earliest examples of genetic engineering was the production of human insulin in bacteria. By inserting the human insulin gene into bacterial cells, scientists were able to create a reliable and affordable source of this life-saving hormone for people with diabetes. Genetic engineering has revolutionized agriculture, leading to the development of crops that are resistant to pests, herbicides, or harsh environmental conditions. It has also found applications in bioremediation, using genetically modified organisms to clean up pollutants and toxins. However, genetic engineering is not without its controversies. Concerns about the safety of genetically modified foods, the potential for unintended consequences, and the ethical implications of manipulating life itself have fueled ongoing debates. Despite these concerns, genetic engineering continues to advance at a rapid pace, driven by the promise of groundbreaking discoveries and life-changing applications. Section 7: CRISPR, the gene editing revolution. In 2012, a revolutionary gene editing tool known as CRISPR-Cas9 emerged, sending ripples of excitement and apprehension through the scientific community. CRISPR, short for clustered regularly interspaced short palindromic repeats, allows scientists to edit DNA with unprecedented precision, akin to a molecular scalpel. This groundbreaking technology, adapted from a bacterial defense mechanism against viruses, has the potential to cure genetic diseases, enhance crop yields, and even reshape the future of our species. CRISPR has already shown promise in treating genetic diseases such as cystic fibrosis, sickle cell anemia, and muscular dystrophy. In preclinical trials, CRISPR has been used to correct genetic defects in human embryos, raising ethical concerns about designer babies and the potential for unintended consequences. The CRISPR revolution is still in its infancy, but its potential is vast and its implications profound. As scientists continue to refine and explore the capabilities of this powerful tool, we are entering an era where the line between science fiction and reality becomes increasingly blurred. Section 8: The ethical frontier, navigating the promises and perils of genetic engineering. The rapid advancement of genetic technologies, particularly CRISPR, has thrust us into uncharted ethical territory. The ability to manipulate the very essence of life raises profound questions about our responsibilities, our limits, and the kind of future we want to create. The potential benefits of genetic engineering are undeniable. We have the opportunity to cure devastating diseases, alleviate suffering, and enhance human capabilities. However, these possibilities are accompanied by significant risks and ethical dilemmas. The prospect of designer babies, where parents could select their children's traits, raises concerns about genetic inequality and the commodification of human life. Unintended consequences of gene editing, such as off-target mutations or ecological imbalances, could have far-reaching and irreversible consequences. Navigating this complex landscape requires careful consideration, open dialogue, and robust ethical frameworks. We must proceed with caution, ensuring that these powerful technologies are used responsibly, equitably, and for the betterment of humanity. Section 9: The future of DNA, from personalized medicine to unraveling the mysteries of life. The journey of DNA from Misha's nuclein to CRISPR has been one of remarkable discovery and relentless innovation. As we delve deeper into the secrets of the genome, the future of DNA research promises to be even more transformative. Personalized medicine tailored to an individual's genetic makeup is poised to revolutionize healthcare. By understanding an individual's genetic predispositions, doctors can develop personalized prevention strategies, early detection methods, and more effective treatments. Beyond medicine, DNA research is shedding light on the mysteries of life itself. By comparing the genomes of different species, scientists are gaining insights into evolution, biodiversity, and the interconnectedness of all living things. The journey of DNA is far from over. It is an ongoing saga of discovery, innovation, and ethical exploration. As we continue to unlock the secrets of our genetic blueprint, we have a responsibility to wield this knowledge wisely, for the benefit of all humankind and the planet we call home.